This is a pleasure for me to come to Oslo. It's such a beautiful place. Thanks for the invitation. This is the first time in the winter that I see snow, so I'm still excited. Hope it snows a little bit later. So I like this weather, especially coming from Barcelona. So I've decided to give my talk more of such a conceptual view of what we have learned over the last couple of years in precision medicine, and I use colorectal cancer as a model. So I don't want to focus in the details. So this is not teaching you about particular targets or novel drugs. It's just more like an overall view of how I think the field should move forward in terms of integrating omics to clinical and research. So this is a paradigm shift in precision medicine with colorectal cancer as a model. So when I first arrived at Vall, we started to do molecular profiling. So I came there because I thought, well, in colorectal cancer, we need to find the drivers like we had at that time in 2010, 2011. In lung cancer, EGFR mutation, you have a targeted drug. And I believe that this would be the case in colorectal cancer as well. So we started originally with this single gene, single biomarker, single drug paradigm. And that's when I arrived there. And the field has moved to a multi-gene, multi-biomarker, multi-drug paradigm. And in the future, I think that we are already living this perspective of how to see a patient and a potential biomarker, which is multi-omic systems biology, and impossible not to mention adaptive immune sort of therapy that we will have in the future. So I will start with the past, because we learn from the mistakes. And this is the original idea that we had. That's the concept. We pre-screened patients to enroll them in early clinical trials with molecular targeted agents. Of course, you need a clinical trial environment to test all these hypotheses for drug biomarker matches. So we started to pre-screen patients, and the idea was originally to pre-screen them. At the time, they were in good shape, fit for a clinical trial. Because it takes time, the turnaround time at that moment in the past was relatively long to have the results of a molecular pre-screening, so you needed time to understand the biomarker. And then when the patient progressed to standard therapy, we selected a target based on the results and followed the patient. So the original idea that we had was, if you have a KRAS mutation, a MEK inhibitor, a BRAF inhibitor, PIK3CA, an inhibitor direct, and so on. So the idea was, because this was how we were living and seeing positive results in other diseases, one test, one drug, the companion diagnostics. And this is also a lot of pressure from the companies, because at that time, that was the way to do drug development. But we had less than 5% response rates in colorectal cancer with all these ideas. So the first question that came to my mind, well, this is the drug that is not working. It's early clinical trials, the drugs are investigational agents, maybe they are not potent enough against the target. And the other point is that, well, what I'm doing here is pre-screening with a sample from archived tissue of the primary, and that may be different from the metastasis that I'm treating now. So this was the second hypothesis. The drug, well, if you compare the response rates in other tumor types, like with the KRAS mutation MEK inhibitor in lung, we didn't see much responses, but there was a clear signal. At least half of the patients had tumor shrinkage. They were benefiting for long term. Not to mention BRAF mutation, BRAF inhibitor in melanoma, responses rates around 60%. So the same drug we were using for colorectal and not working at all. PI3K in breast cancer, also very nice responses. So with a PI3K alpha selective inhibitor, in gastric cancer, for example, again, MET amplification with a targeted agent, spectacular responses. It's hard to find these patients, but when you treat them, spectacular responses, and that was not the case in colorectal cancer. So the next question is the metastasis different from the primary? So I will come back to this question later on, but for the biomarkers that we were enriching patients in clinical trials, KRAS, BRAF, PIK3CA, MET, apparently there is no heterogeneity in the primary metastasis evolution. So it wasn't quite really the main reason for the failure. And so what happened is that the only way to improve is really to a deeper and biological understanding of the disease. And this is what happened in the last 3 years. First, at the genomic level. So we started to understand better colorectal cancer at the genomic level. And then we moved to this new paradigm of multi-gene, multi-marker, and multi-drug for precision medicine. And this all started with the Cancer Genome Atlas. So as you can see here, we realized that, not that we didn't know it before, but there are co-occurring events. These events do not happen in isolation, these potential driver alterations as in any other disease, but in colorectal cancer particularly, there is combination of aberrations. And this is what we learned from the first results of the Cancer Genome Atlas. So don't look at one in isolation, look at one potential driver aberration in the context of other aberrations, right? We started to understand better the dynamics of target evolution. So the classical example here is the BRAF story. So you hit BRAF with a potent drug, but there is a feedback mechanism that activates through EGFR, other pathways. So this also happened, and BRAF is one example, but in many other situations, we started to, again, understand with more deeper profiling of the tumor what was happening. And also another important concept that is clonal selection and temporal heterogeneity. No? So if you have a targeted agent that works fine and well, for example, EGFR in KRAS wild-type population, what happens is that you have a clonal selection and the sensitive cells, they do respond to therapy, and you have tumor shrinkage, but at the time of relapse, the resistant cells dominate the tumor omics. So this was very clear in colorectal cancer. If you compare primary tumors before exposure to anti-EGFR therapies with highly sensitive techniques, and you do the profiling after anti-EGFR therapy, you start to see that all the originally reported resistance mechanisms, they emerge later on. I will have a slide, for example, KRAS, NRAS, BRAF, PIK3CA mutations, EGFR mutations, all aberrations that were defined as upfront primary resistance to anti-EGFR, they end up reappearing after exposure to anti-EGFR agents. So what happens is that you treat a population that you believe that is all wild type upfront for all these aberrations. So this is the responsive population. But at the time of relapse, all these minor clones that were hidden here, they start emerging. This is something that we learned in colorectal cancer that was something unique and could be translated also to other diseases. So we now move to what we call a genomically stratified or multi-categorical model. And it's dynamic, it's context dependent. you do the profiling, you find an alteration, you give one or multiple drugs. But importantly, you try to do this as frequent as possible, or when you believe there is there is a shift in the clonals involved in the disease. So we might have a biopsy or a profiling done before standard of care therapy, after standard of care therapy, at the time of relapse to an experimental agent. And more we started to add, because it is just impossible to get biopsies, longitudinal biopsies, frequent biopsies for all patients. So circulating free DNA started really to come out as a good source, at least at the genomics level, to capture the aberrations that were happening in the tumor. So the paradigm here is now you have to take into consideration the context and what the patient received in the past, and if it's chemotherapy only, if it's targeted therapy, and you never consider one biomarker in isolation, but multiple biomarkers. And it's very hard to see one drug working in isolation. You need, in many cases, combination of targeted agents. So these are examples here. You have a quadruple wild-type population that is probably responsive upfront to anti-EGFR agents. A BRAF requires multiple drugs for targeting in colorectal cancer. In the setting of acquired resistance to anti-EGFR, if you have ERBB2 amplification, you respond to a combination of targeted agents. In the setting of acquired resistance plus BRAF wild-type, emergence of EGFR mutation, you give a second-generation, more potent anti-EGFR therapy. So it is not simple. It is starting to get more complex, because you need combination of markers and settings to define the best therapy. So this is just one example for resistance to anti-EGFR therapy, acquired or secondary resistance, all the aberrations that can happen, and the multiple clinical trials that were investigating agents specifically directed to these therapies. So now and more, it is what we see in clinical trials is, again, taking into consideration previous therapies in order to define the subsequent therapy in colorectal cancer. And this is, again, just one example. In breast, in lung, and other diseases, it's happening exactly the same way. Combination of biomarkers up front, in the acquired resistance, new biopsies, new sampling, you give agents accordingly. And then for the first time, we started to see response rates that are reaching 30%, 40% in colorectal cancer with matched targeted therapies. So it is working. It is not yet there as in other diseases, but it's starting to work in its early days, because we have the results of only some clinical trials, and this is what I'm showing here. The positive examples. This is what I was telling about the EGFR RAS wild-type population with second-generation anti-EGFR. You see all the responses are clustered here in the BRAF or the KRAS BRAF right-type population. You even have responses in the ones that are EGFR mutated. The ERBB2 KRAS wild-type population with a double hit to HER2. This is amazing response rate and never seen before. It's extremely hard to find this patient. They represent 5% of the population in anti-EGFR refractory, but if you find the aberration, this is a driver, and you have potent drugs, so the drugs are working. The BRAF story that you are well familiar, the responses that we see with combination of targeted agents around 30–40%, and long-term clinical benefit in a population that we know is extremely aggressive tumors and poor prognosis. And we were also able to, by doing more deeper analysis of the omics profile identified these very rare aberrations that were sometimes described in other tumor types, also in colorectal cancer. We started to see RET, ALK fusions, but also this is a TRK fusions and TRK1 fusion in colorectal cancer. It's extremely rare. It's less than 1% of the population. But if you find it, this is an oncogenic dependency, yes or yes. So you have very nice responses. So it started to get complex because it's not simple gene mutation. That's the first time that we see that other aberrations are playing a role. And also beta-catenin for the first time with non-canonical aberrations in the pathway. Also with a match-targeted therapy, we started to see, this is preclinical work, but we already have case reports in the literature of responses to this novel agent. So what happened is both development of new targeted drugs for specific rare aberrations in colorectal cancer. But it is still not enough. We still need to improve the results further. And what we believe, and this is from now on conceptual, but I think I will convince you that that's the case, we need a deeper understanding at other omics levels of the disease. So adding transcriptomic and immune level will probably help us understand the failures and what to do next to improve the response rates. So then is when we move to the multi-omics and systems biology, adaptive immune drugs. So now the molecular or the pathologist is not the only one helping you make sense because of the amount of the data that you might be able to generate with omics. You need a computational bioinformatician, computational biologist, oncologist helping you prioritize therapies and understand the multi-omics landscape, especially because of the immune therapies that you are well aware of. So this is just to show that we were able to really identify intrinsic subtypes of colorectal cancer that we believe are easy to translate to clinical practice because of the robustness of these results, and we have multiple series now with validation of these results, and we have 4 subtypes, one that we call MSI immune, mainly MSI population, with very high immune infiltration and activation, and this is one bottleneck, a clear bottleneck. The canonical subtype with EGFR addiction, Wnt, MYC pathway aberrations. You have the metabolic subtype that is highly enriched for KRAS mutation and metabolic deregulation, and the mesenchymal, where the stroma is really dominant feature in the tumor with activation of immune suppressive agents like TGF-beta and a very poor outcome. So I'm going to show you just a couple of examples of how we believe that adding transcriptomics and immune subtyping in colorectal cancer can help us in the future to guide therapy. So very nice publication last week on first clear link of KRAS aberrations, mutation plus copy number changes in KRAS, leading to a metabolic reprogramming and therapeutic suitability. This was already described superficially in other diseases, so now we have an explanation why we see such a metabolic deregulation in the KRAS mutated subtype. That so far, as you saw, it has not been really a targetable aberration in colorectal cancer. For KRAS, which is 40% of the population, we still have no particular targeted agent. And maybe we need to add the understanding of the metabolic deregulation in KRAS to stratify patients. So it's not all KRAS mutated, but there is a subset that is really, in this publication we see that this is a subset that has copy number changes in KRAS on top of KRAS mutation. That leads to a metabolic deregulation. And that's probably one bottleneck. And in lung cancer, we have seen in preclinical models exactly the same. And so the field is moving towards not only MEK targeting in KRAS, but also understanding new susceptibilities. DNA damage related to a metabolic enzyme inhibitor, repurposing of chemotherapies that were used in the past and didn't work in the overall colorectal cancer population, that maybe in this setting they start work. So we will probably go back and see previous failures and try to understand a particular subgroup that might be responsive. And I think that the most important is the idea of the immune subtyping of the disease. So it's not only the MSI population in colorectal cancer, macrosatellite unstable population in colorectal cancer that is immune infiltrated and immune addicted. You see a wide range of immune alterations in colorectal cancer in the MSS population that might be hypermutated as well or not. And what we know right now is that, again, defective mismatch repair, hypermutation, you have a clear metabolic, you have a clear immune infiltration with all the agents that are activating the immune systems. PD-L1 plays a major role here. But you also have an immune tolerant subtype of colorectal cancer. That's the mesenchymal subtype. And this subtype is related in part to inflammation, chronic inflammation, colitis-related colorectal cancer, TGF-beta, complement. The innate immune system is all there in the tumor. Is not active because you have all these immune suppressive molecules, chemokines, and cells blocking the immune systems from the activity against the cancer cell, but they are there. They are just not able to attack the cancer. So this is, again, very strong evidence for multiple experiments and immune phenotyping. And you have also an immune ignorant subtype, which is, in essence, the CMS2 and the epithelial CMS2, canonical and metabolic. These tumors, we need a different strategy if you want to target with immune therapies this group. We already have a proof of concept, the MSI PD-1 blockage. So in blue here, you see the mismatch repair deficient population that almost all patients have tumor shrinkage. Again, very rare population. We are talking about less than 5% of the metastatic population. But if you find it, it's the amazing responses that you see in the clinics. But we believe that we can expand this. And there isn't hypermutated or a population that, well, what is the cutoff for hypermutation, but maybe a population that with high presentation of neoantigens, neoepitopes that might be sensitive to PDA blockage alone. But there is also a much larger population that we know has immune infiltration with repressive or inhibitory cells. And the strategy here would be combination of immune stimulatory drugs and inhibitors of immune suppression. A single agent will not work. You need combinations. So this will all complicate when we start to add on top of these vaccines, T-cell directed or related therapy in colorectal cancer. So this can be like expanded to all potential subtypes of the disease. So my view is that we originally had the past, we have to forget. Again, this is not working. We learn from the mistakes, but right now this is what we are living, a multi-gene, multi-biomarker, multi-drug. And because this is genomic related, our view is a clonal perspective of the disease. So you have targeted agents that target specific clones, and they evolve over time. So what we learn about co-occurring alterations, dynamics of target inhibition, the clonal selection, the temporal heterogeneity, and the oncogenic dependencies. This is what we are living now, and this will stay. This will not. This is not gonna change for a long time, and so we will learn how to treat not the cancer, but the tumor evolution, the clonal evolution. And this is a major driver, a major shift in the way that we think about the disease. We are treating the clones that are evolving. We are treating the disease that is able to evolve, and we have to see this in a different way. But in the future, we will have a stromal and immune perspective of the disease. And then the microenvironment plays a major role. We will have to understand better the microenvironment, what is happening in the microenvironment in terms of immune activating, immune suppressive cells. And also, what I think is that we will learn more and more that there is a spatial heterogeneity in colorectal cancer. There is intermetastatic heterogeneity, not only at the clonal genomic level, but also stromal and immune level. So it will be, it's probably going to require a lot of efforts from the community to understand really what is the intermetastatic heterogeneity. But if we focus on microenvironment of intermetastatic or between metastasis changes, we will probably learn even more about the disease and how to move it forward in terms of targeted therapies. So my last 5 slides are what I think that how to make precision medicine a reality. So it is very hard to implement molecular pre-screening on a clinical setting and to treat patients in clinical trials. The challenges there are huge. So we have to be aware that this is not a simple understanding, especially because in many cases you need the availability of multiple different targeted agents. So it's not only one. It's very hard to find this. Because of the rarity of the events, it's very hard to envision that you can run a trial or select a population who will be successful on a single institution basis. So you need collaborations, you need multi-institutional trials, you need extensive profiling, you need access to multiple targeted drugs. And this also, the only option is really to go for innovative clinical trials. And so the profiling has to be done at multiple time points, and we understand the difficulties there, but this is something that is critical. We will change the way we treat if we look at the dynamics of the clonal and the tumor evolution over time. So just to give you examples of these clinical trial designs that we have currently and we will have in the future, that I believe this plays a major role for precision medicine. So the basket trial, you have one aberration, you have one drug, and you treat all potential tumor types. Imagine the NTRK fusion that I was describing that happens in less than 1% of the tumors, but in many different solid tumors. So you need cohorts for different diseases, and you see the activity, you compare the activity in one disease with others, and this is one way to go. But it has limitations because you have to do the profiling here. Imagine here in lung cancer, to have to profile at least 100 patients to find 2 or 3 that are eligible for the clinical trial. So the limitations here is that 98 of your patients that you profile are just not eligible for the particular drug. So it is not, from a patient perspective, this is not a good clinical trial to be enrolled. If you have the aberration, perfect, but if not, so in order to expand this, we are more and more moving to this umbrella type of clinical trial, where you do the profiling up front, and you have an option for every potential aberration that you find. And you even have a non-match group that you do biomarker analysis and you might give immune therapies, for example, if you don't clearly find, or immune plus chemotherapy combinations. And then here, you can either make it randomized or a single arm. But again, all patients are potentially eligible. The problem here with this type of trial is that you need access to all the drugs. And so you hear Genentech, you see Pfizer, you see AstraZeneca drugs. So it is hard to convince all these pharma companies to collaborate in one single arm. But more and more they understand that this is the only way to go. So we have many, many more examples of this type of trial, umbrella trial, that you really give your patient, from a clinical perspective, a very good option, and really a personalized therapy in many cases. And what I also like is this window of opportunity or design. What is this? This is a situation what usually happens in colorectal cancer, for example, that you give therapy up front with standard of care, the patient responds to treatment, and then you wanna be less aggressive with therapy, or you wanna just give a chemo break because you have the disease under control, so you give a chemo break, and in this maintenance phase, that is around 50% of the colorectal population that you are able to make a treatment break. You use the information from molecular pre-screening up front to guide therapy here. BRAF mutation, you randomize here, imagine HER-BB2, alteration to randomize, and you have a no biomarker arm with immune therapy. And an important concept here, this is the adaptive design. So adaptive because I change the biomarker. I change the drug because I know that the drug, for example, is not working, or I add a new biomarker. And this is interesting because it's really from a patient perspective, it's in a situation where you don't need to be aggressive. You can do experimental therapies without major impact in the evolution of the disease. And this is all, again, within a clinical trial scenario. You are careful and you follow up the patient very closely. So I think that in the future, and that's why I think really that in the future we will be more adaptive with therapy, because we will understand what is changing in the dynamics of the tumor, but we need to adapt as well the clinical trials. You may need to add a new pathology analysis looking at specific immune markers. You need to expand your profiling not only to BRAF, but BRAF plus RNF43 mutation. You may change the way you look at the efficacy of the drug, if it's not only response rate with the standard criteria, but maybe an immune-related response criteria. You adapt the drugs. If the drugs are not working, you should try combinations. And of course, the clinical outcome in terms of quality of life and patient-reported outcomes. So this is my vision. I think that this is it has been a paradigm shift in colorectal cancer. It is happening, and I think that more and more the clinicians will need to understand omics, and will need to collaborate with preclinical and translational investigators to really comprehensively characterize a tumor and give the best option. And this is not an easy undertaking. It's going to take time, but I think that you have the resources and the expertise to do this as well. So I would like to thank people from my group. I have a group in Vall called Oncology Data Science, and what we do is for all patients that undergo molecular pre-screening, it's around 1,000 patients every year right now that we do next-generation sequencing. Only a third of them and end up being enrolled in a clinical trial with a match-targeted agent. Even though I would say that we find something that is potentially targetable in 80% of the patients, we only enroll 30% of them in clinical trials, so there is a major loss in terms of evaluation of predictive markers. But we capture the clinical information of these patients that undergo molecular pre-screening and try to understand the prognosis and compare, identify new biomarkers and do all the correlative analysis. So I have a group that is really integrating molecular data with clinical outcomes. And we are learning from you that you have a spectacular team that does clinical curation, and this is just the beginning. And Giuseppe for his help there, and Sage, that it still makes me like an exciting. I learned from them that you have to be excited with the next step, and I think that the immune field is just exploding right now, and it's not because of the. It's not only because of better understanding of the immune subtype of a disease, but also because we have better drugs as never before, immune therapies. So there is also at Sage Bionetworks a movement to, in cancer, prioritize biomarker discovery in the immune field, and this is what I'm digging now and trying to understand more and more. And it will be exciting years in the future with a combination of immune and targeted and chemotherapies. So thanks for your patience, and I'm happy to take questions.
Thank you very much, Dr. Dienstmann. Could we sit up on the stage, on the chairs there, if you take the one? Please bring some water. And for the audience, please submit questions on Slido if you haven't already done so. And hopefully we can get a good discussion.
Oops.
We are tall in Norway.
Yeah.
So it was very, very elegant, the design that you're putting together in Barcelona. My kind of first worry was how on earth are you gonna afford this for the vast majority of patients? I mean, even in Norway, where we have a lot of oil, we struggle with how we're gonna pay for all this. What are your thoughts about the cost of this in the future? How are you gonna afford it?
Yeah, right now what we do is we do molecular pre-screening for patients that are eligible for clinical trials. So this is a biased population. So it's not upfront for every patient. It depends on the disease type. You might do this upfront because you have clinical trials that you can guide therapy from the early beginnings. But in other cases, when it's a more refractory population, the clinical performance and patient's interest and the physician engaged with the patient, and that's where you really see an increase in molecular pre-screening. So right now, we have molecular pre-screening that is largely financed by the institution. And this is through collaborations with pharma companies. So if a pharma company decides to run a clinical trial there is part of the budget that is related to pre-screening. We have a calculation. Imagine they are looking for an aberration that it happens in 5% of the patients. And so we show our data, we show our results of molecular pre-screening, and we say, well, we have to, if you want to have 5 patients enrolled in the trial, this is the total cost for molecular pre-screening to identify these 5 patients. So how can you help us? And that's usually not a major issue. So that's that's usually welcomed from the pharma perspective because they understand the value of all this. And in some cases, you have to send samples for central screening, and then the cost is 100% covered by the companies. I think that the best option is really to do in-house screening, because then you can control your results, you generate your results, you learn from your results, and from your population, and you control it more. And yeah, that's basically is. And then there is also a large international consortiums with financing from the European Committee that are also helping us add these new biomarkers. When you move away from the omics, genomics only, when you move away from only looking at mutations and fusions, copy number changes, all these new biomarkers at the transcriptomic and immune level, you can only afford this through international efforts with finances from big, really institutional and academic groups.
So on one hand, you have good collaboration with the pharmaceutical companies to finance part of this genomic analysis that you're doing.
Mm-hmm.
On the other hand, are you also building up kind of the capacity in around Spain to do these kind of comprehensive genomic analysis in other hospitals? Or
we have the same way that is happening in France. We are doing a centralized laboratory analysis for many. It's much harder to and more expensive to run analysis in independent labs across the country than really centralize it, and because of the expertise. And the more samples that you profile, the more expertise you have. You can also design your panels more efficiently, more cost effectively, and it's much cheaper. Also, a good point is that all these molecular pre-screening programs have to be very adaptive and dynamic, because every 6 months or so there is a new marker that comes out, a new mutation that you need to add to your panel. So you have to control your own panels. You have to design your own panels and run your analysis, and not use commercially available solutions that are not as flexible as you may need at the clinic. So the molecular pathology lab is a fundamental step in this. And they have to, again, work non-stop to be as flexible as possible, and so on. And in many cases, like for circulating tumor DNA analysis, what we do right now is collaborations with biotechs that have new technologies. So when it's early days, when you still don't have the capacity to run it yourself, you start with a collaboration with a biotech, with a new company, and then you learn and you design your own panel and you adapt, because they are usually they want to they have a market that is very clear, and you want to expand on top of that. So that's another option.
Good. So we're going to take a question from the audience, from Professor Skotheim. How robust is the CMS subtyping that is across multiple biopsies from the same tumor? And can it be misdriven by the amount of stromal and cancer cells?
Well, unfortunately, we don't have a lot of data in terms of multiple sampling of the same primary tumor to see how consistent the subtyping is, or how concordant it is. The only thing I can tell is that we did look at some data, and apparently the tumor purity in the sample is not a major driver of the classification. So sometimes there is in a lesion you have regions with more immune infiltration, with more stromal infiltration, and this is probably having an influence. But when we when we take the invasive margin that you know that has more immune infiltration, or you take the tumor center that has less immune infiltration, you still see more or less the same gene expression classification, because that's the field effect that is going on in the tumor. So this works. This is a case for the primary colorectal cancer. The problem is that in the metastasis, the classifiers that we developed, they do not work. So in metastasis, because the stroma is different from the primary, because you have, imagine a liver metastasis, so the microenvironment is different. Not that you don't have immune or stromal infiltration, but it's a different context. So you have contamination in the gene expression level of cells that are different from the primary. So you start to see major shifts from primary to metastasis that we believe are not 100% biology. They may be related to a technical issue, accuracy of your classifier that was originally developed for the primary colorectal cancer tissue. So every time we try to translate the classifier to other metastases, you see shifts, and they are all explained by the stromal changes and the immune changes. But in the primary, again, I'm talking about 5 samples that we have in-house that we looked specifically at different regions, and it's more or less the same. I'm talking about, again, I have two or three examples that I was able to classify. So I think that the stroma and the immune are major determinants of the classification, but it really makes a difference when you look at the metastasis. The primary, it's still missing.
Arnett?
Thank you for an excellent talk. I just wondered, there are a certain percentage that you are not able to classify using the CMS. I mean, couldn't that be sort of 2 different CMS that more or less grows together? So indeed, there were heterogeneity to start.
So when we did explore the unclassified samples, we saw that really most of them had a mixed phenotype. They had very strong signals of one and weaker signals of another. So it's not strong enough to make it like a strong statement. It's all about cutoffs again. In terms of gene expression classification, you reduce your cutoff, you are able to give a label. But if you stick really to a very accurate cutoff for prediction, there are samples that have a mixed phenotype. And I think that this is biology real. So this is not we are just picking a snapshot of the tumor at that moment. We are not picking the dynamics that is happening. So because they are in fact a translation from the genomic changes or the epigenomic changes that are happening. And this is happening constantly in the tumor as they evolve. So we saw, interestingly, in some cases, tumors that were originally classified as one epithelial type, in the metastasis after multiple chemotherapies, the tumor had more mutations, TP53 mutations and so on, and it acquired a more immune-related classification. And I think that this is probably going to happen. Shifts that are related to the aggressive chemotherapy that you give during the course of the disease. So, yeah.
Well, that's kind of one of my worries, that in colorectal cancer, the kind of standard of care is to give these kind of multi-drug, multi-chemotherapy cocktails where you hit very broadly. And are you going to kind of introduce your either immunotherapy or your targeted agents after all this has failed, or are you kind of implementing and trying to get it in first line? How do you do this?
So in colorectal cancer, we are doing for the MSI population or the hypermutated population, the signal is so strong up front, and these patients have a relatively relatively poor prognosis as compared to the other tumors in the metastatic setting. So as early as possible, you give the immune therapy because you don't want to lose the patient if it fails to standard chemotherapy. So the trials are moving to immune therapy in this population as first line. So we already have phase 2 clinical trials, and I think that this is going to be approved very soon. MSI, first line immune therapy. But for the other tumors that we believe are also responsive, the mesenchymal population that is immune suppressive microenvironment, so we are starting a trial now with a combination of TGF-beta receptor inhibitor plus OX40 agonist, that's an immunostimulatory drug, in second line, right after failure to first line therapy. That's where you have the strongest signal of activity, first line, so you are still giving the patient the opportunity to respond to standard of care first line chemotherapy, and upon progression, you give the targeted agents. That's because the rationale and the preclinical and early clinical evidence to move this to first line is still very poor. But we don't leave it to much later stages when you don't understand what is going on. In lung cancer, for example, with immune therapies, there has been some indirect evidence from clinical trials that if you give it in the first line setting, in the second or in the third, fourth line settings, you still have more or less the same response rate. That's with PD-1 blockade alone. So patients that received aggressive chemotherapy and that might have led to a more immunogenic microenvironment because of immunogenic cell death or because of aggressive chemotherapy, they are not responding more to PD-1 blockade alone. But we don't know in colorectal cancer whether this is the case. But the message is that I think that it's a mistake to leave it to very late stages. Right now, we know what the PD-1 agents by itself give in terms of response rates. So right now, we are leaving the combination therapy era, and then I think that as early as possible, you have to give.
Do you think there is any indication that you can use chemotherapy to kind of ignite or to fire up the immune system, or is that more kind of a theoretical hope that we have?
Well, that's theoretical, and in colorectal cancer, there is a lot of preclinical research also with oxaliplatin, for example, leading to immunogenic cell death and releasing factors in the microenvironment that stimulate the immune system. So there are clinical trials specifically looking at whether combination of oxaliplatin-based chemo plus bevacizumab to reduce the interstitial pressure plus PD-1 blockade will work. So I'm not quite convinced that this is like in an unselected population. I don't think that this is the case. But I think that there is one missing point here, is that's inhibitors of the immune suppressive microenvironment. So it's not only PD-1 blockade. You need something to block the immune suppression that is happening in colorectal cancer, or maybe immunostimulatory drugs, whatever immune stimulatory drugs you have. So this is reality.
Good. Professor Myklebust has asked the question: how do you make available the outcome of sub-trials that do not work?
So we are, well, right now it's very unusual to have a clinical trial that is never reported. We pay a lot of attention to abstracts presentations in conferences, and these, because of the amount of, the number of targeted agents available, it's very hard to see that all of them will work and will be acceptable for a publication. So we have to pay attention to this as well. But we are launching, and we, in combination with other groups like here in Europe, consortium that that is focused on case reports online that you submit your case, completely de-identified, obviously, and for every patient that you believe you are giving the best match therapy in terms of precision therapy guided by molecular aberration, you can submit in this website the results. So I gave to this patient with AKT1 inhibitors AKT inhibitor, and I did not have a response. And this is the profiling of the tumor. So more and more, the case reports are going to play a role as well for us to understand. And this is all about consortium and data sharing. And I think that this is, in the US, they have the Cancer Link tool. So all it's an ASCO program. So many community hospitals that do molecular profiling and guided therapy according to molecular pre-screening, they are obliged to submit the results of any potential match therapy intervention to this big database. So it will become available in the future. So I think that the clinicians will be able to look for answers in the web. So I have a patient that I have one aberration. Is there any response rate reported in the literature with this, with a patient that has a similar profile, and what worked, what didn't work? Yeah. So the ones that didn't work are the most critical ones for us to understand.
There will be a negative kind of publication bias. These things will kind of not be published on our kind of conventional PubMed, but exactly. You need to kind of look up these websites. So another question, are chromatin changes likely to inform treatment choices in the future?
Well, in many diseases, rare diseases, we started to see, like in sarcomas and with this new epigenetic therapy that are more directed to a specific target, we understand the potential aberration that is reflecting an activated epigenetic context, or and not activated, but I would say an epigenetic dependency. I think that this is going to change. The epigenetics is another field that we don't understand a lot, but I think this is going to change the way we treat many cancers in the future, especially combinations with immune therapy as priming of efficacy of immune therapy. But also in colorectal cancer, I think that combination with chemotherapy has to be revisited as well with chromatin. So I don't think that really a specific chromatin change is going to be used as a biomarker, but we will infer what is happening at the epigenomic level, and then we will probably expand our epigenetic drugs in the future. But this is really early days. For the epigenetics, this is early days, especially in solid tumors. This is early days.
Yeah.
I have a simple question. Here in Norway, the stage 2 patients, they are offered surgery. If one uses the CMS classification and you find a patient with CMS 4, and there is no trial available, what would you suggest one should do?
So I suggest that you should design a trial because as we have strong evidence that CMS4 is a poor prognostic marker, we also have some early data showing that that's not predictive of efficacy of chemotherapy. So having a poor prognosis doesn't mean that you will respond more to chemotherapy. So with the chemotherapy that we have right now for stage 2, 5-FU-based chemotherapy, that's probably not enough. So I would probably I would wait for the results of the clinical trials going on in metastatic setting, seeing combinations with immune therapies or something. Yeah, I would probably, or I would have a new hit of a potential combination with chemotherapy, an agent that I believe, maybe epigenetic therapy combination with chemotherapy, that I think is particularly active in a mesenchymal situation. And then I would design a trial, a proof of concept trial. But, right now, I would, if I had this patient in my hand, and if it's a young, fit patient, I would favor chemotherapy, but not based on preclinical or clinical evidence of this being predictive of response to chemotherapy, but I would try to give the best to my patient.
Could it be that it had a lack of CDX2? Would that be like a simple test to add, and then just the chemotherapy? Or?
So we did analyze the CDX2 in the context of the subtypes, and what we see is that it is really identifying a population in CMS1 that has worse prognosis.
Okay.
Okay. So the problem with that publication is that it is the work in the validation, not in the training, but the work in the validation didn't take into account MSI as a marker of good, poor prognosis. And what we see is an enrichment for CDX alteration in the CMS1. So at the end of the day, we're identifying a rare population within the CMS1 group that has very poor outcome. And it's not enough yet to move.
Well, thank you very much. It was a very inspiring lecture that you had, and we look forward to see more of your results. And we also hope that we can collaborate in the future. Although we're up in the north and there's a lot of snow, we still kind of like to think scientifically.
Good.
So
even
thank you very
more
much.
Than ourselves, I believe.
Taking a long trip. Thank you.